everybody. Today we're going to start with our first lab for the microbiology lab. Today we're going to prepare media and sterilize and perform aseptic techniques. The learning outcomes of this lab 1 are: first, to describe the different types of culture media and their composition. Second, to demonstrate skills in the preparation of agar, agar dip, and agar slime. Third, to describe the importance of aseptic techniques in microbiology. As you can see over here in this slide is the artificial media being prepared to support the growth of bacteria or fungi in microbiology lab. This is very important for the microbiologists to study the biology of the bacteria or fungi. In the environment, bacteria and fungi are mixed together in the ecology. Therefore, an agar, solidified agar, is very important for us to isolate the bacteria or fungi, as you can see from this petri dish. You can see there are a variety of bacteria growing in the agar plate. Some colony looks yellowish, some grayish, whitish, some even have red color colonies. And this represent different type of bacteria in the ecology. This single colony of bacteria growing on the agar represent a single species of population of bacteria. Therefore, from this mixed culture of bacteria, we're going to pick up this single colony and restrict it onto a fresh agar plate, and therefore we're able to get the pure culture of those isolated bacteria. There are a variety of media being prepared in the microbiology lab. As you can see from the figure over here, there are slant or deep, and then deep agar. Those are solidified agar, and we have a liquid media of broth and also broth with the additional of Duhan tube and also agar plate. As you can see from here, culture media can be differentiated based on several criteria. We can group the type of culture media based on physical properties, the chemical constituent, and function. The group of culture media based on physical properties, as you can see from the figure itself, it can be in liquid form, semi-solid, and solid. For the chemical constituent, we can have defined or synthetic or complex. And based on the function of the culture media, we can group them as supportive, enriched, selective, and differential. Let's look at the physical classification of culture media. In a liquid media, we can culture the bacteria or fungi in a larger number of bacteria and fungi. However, if we use the liquid media to isolate a single population of species of bacteria, we are unable to do it in the liquid media. In the solid media that we're going to prepare in this lab experiment, we're going to prepare the agar slant, agar dip, and the petri dish. So that is the type of the solid media that we're going to prepare for the grow isolating a single colony of bacteria, and also for the maintenance of the bacteria culture in the agar slant and also in the agar deep. The bacteria culture in the petri dish, we can maintain it in relatively about 2 to 3 weeks until the agar being dried out. However, the agar slant and agar deep, we're able to maintain the bacteria or fungi culture more than 1 month or 2 months of store in the refrigerator. In a semi-solid media is a type of culture media that have the property between the liquid and solid media. It's a little bit like a jelly-like due to its low percentage of solidifier such as agar being added into it. Usually it may contain about 0.4% or 0.5%, where for the solid agar media, usually the agar being percentage of agar being added are between 1% to 1.5%. As you can see from here, this is a type of gelatin agar that being used for testing of mortality of bacteria in the semi-solid egg media. Let's look at the type of media that can be grouped depending on the chemical constituent of the culture media. We can group media based on the chemical constituent as either defined or synthetic media and non-defined or complex non-synthetic media. In the defined or synthetic media, all of its component and concentration are known. As you can see from the table, all those chemical constituents such as sodium nitrate, potassium hydrogen phosphate, all those type of chemical constituent being added into the media to support the growth of bacteria, fungi, or algae. In comparison, the complex media contain some ingredients of unknown composition or and or concentration. As you can see, there are complex ingredients being added such as peptone, beef extract, being added, and also the pancreatic digest of casein. All these are complex, unknown chemical constituent of a nutrient that are known of its chemical constituent. Therefore, this is what we define a complex media. Classification based on the functional type. First, we will see the supportive or general purpose media. This is the type of general media being used to grow many type of microorganism in the microbiology lab. Example of general purpose media being used in the microbiology lab, such as the tryptic soy agar, the nutrient agar, and also the LB agar or Luria betani agar. This type of media able to support all these bacteria, fungi, algae on the agar. In comparison, the enriched media are commonly used to enhance the growth of those fastidious type of microorganism that are not able to grow on the general purpose agar. Extra notes that you may take is not all microbacteria or fungi can be grown on the nutrient culture media that we have in the microbiology lab. There are more type of bacteria or fungi still not being isolated on the artificial culture media because all these bacteria or fungi require certain type of nutrient that need to be added into the culture media before they're able to grow on this artificial media. For the enriched media is to usually supplemented with blood or other special nutrient that enhance the growth of those fastidious bacteria or fungi. For example, the blood agar usually enable the microbiologists to isolate those pathogenic bacteria or fungi that usually will not able to grow on the type of general media, culture media. Next is the selective media that usually the microbiologists are going to use to favor the growth of some bacteria or fungi and inhibit the growth of the other bacteria or fungi. The example of this type of selective media is the McConkey agar. It selects for gram-negative bacteria due to the presence of bile salts and crystal violet. So therefore, this type of selective media is being used if we want to isolate certain or specific bacteria that we want to isolate in the lab. And McConkey agar being commonly used in the diagnostic microbiology lab in the hospital due to gram-negative bacteria are usually cause the pathogenic disease in human. Macaulay agar also can be a differential media. It can differentiate between the lactose fermenter and also the non-lactose fermenter. For the lactose fermenter, you see the color changes of the agar from red color into the yellowish color, where the non-lactose fermenter will not able to show any color changes on the agar. The function of differential media is to distinguish between the different group of bacteria or fungi based on their biological characteristics. Here, there are some examples of mechanism of selective and differential media being used in the lab. The blood agar can be enriched and differential because in the blood agar it contains those nutrients that are necessary to enhance the growth of the pathogenic bacteria in our blood system. It can be support the growth of many fastidious bacteria, and the blood agar will able to show those type of bacteria that able to hemolyze red blood cell and bacteria that unable to hemolyze red blood cell. We call that beta hemolysis. If there are appearance of complete hemolysis of RBC in the blood agar. In the iosine methylene blue, it can be selective and differential because there are addition of dyes such as iosine Y and methylene blue that inhibit the growth of gram-positive bacteria. Therefore, only the gram-negative bacteria able to grow on the EMB agar. So depending on the species of the gram-negative bacteria, for example, the E. coli, we're going to produce a distinct colonies on this EMB agar, such as it going to produce green metallic sheen colonies on this EMB agar. You may read all these different type of functional type of media in the manual of the culture media. Salt also being added onto the mannitol salt agar to inhibit those bacteria that unable to grow on high osmotic pressure, such as the 7.5% of sodium chloride. So this can be useful to isolate staphylococci in this type of agar. So to read more about the type of culture media and the requirement of the functional growth, I suggest you to read the DIFCO and BBI Manual, Manual of Microbiological Culture Media, second edition. The link of the manual can be obtained from the link below. In the history, try to read more about the history of microbiology and the culture media in page 3, the microorganism growth requirement, functional type of culture media, the culture media ingredient, and media sterilization. For example, this is the common workflow of food testing for the isolation and identification of salmonella in meat and meat product in the diagnostic lab. Different type of culture media being used in pre-enrichment, the selective enrichment, and identification, presumptive identification of salmonella in using different type of agar. It depending on the requirement of different regulatory institutions such as FDA or USDA. It requires the pre-enrichment in the certain type of culture media, and based on the presumptive identification, the confirmatory identification will be carried out by using the biochemical test, the molecular testing, or the serological test. After we have prepared all these media, we need to sterilize the media in order for us to kill all those microorganisms in the prepared media in the lab so that it will not contaminate in the isolation of the bacteria and also the fungi that we want to isolate. Usually the media are being sterilized by using autoclaving method or membrane filtration techniques. In autoclaving, we usually sterilize the media or plasticware or glassware at 121 degrees Celsius at 15 pounds of pressure for 15 minutes. For the home experiment, I've suggested you to use the pressure cooker, as this pressure cooker will have the same temperature and pressure that require in the autoclaving method of sterilization. This enables us to kill all those bacteria and fungi and the anospores of the bacteria. This method will ensure that no microorganism will able to grow after the sterilization. Usually this type of prepared media, we can store it for very long period of time because there are no microorganisms alive in the media. Sometimes we use membrane filtration for those culture media that contain heat sensitive or heat label ingredient added into the culture media, such as the red blood cell, vitamins, antibiotics being filtered through membrane before added into the culture media. In the membrane filtration, we use a filter with a pore size of not greater than 0.22 micron, or by using the 0.45 micron filter. Other sterilization method can be used, such as, but this we're going to have a less effective method of sterilization compared to autoclaving or membrane filtration method, such as dry heat at 160 degrees Celsius to 170 degrees Celsius for 2 hours. This dry heat method are usually used to sterilize glassware, as the glass material able to withstand at this 160 degrees Celsius to 170 degrees Celsius for 2 hours. So this can be done at home by using your oven that usually used for baking your raya cookies or grilling your chicken in the oven. Other sterilization methods, such as incineration, usually used, such as the direct flaming, that will kill most of the microorganism and the endospore bacteria and fungi. Incineration usually used, or direct flaming being used to sterilize our inoculation loop or needle. Less effective methods, such as boiling, that can be performed safely at home, is to boil your glassware or plasticware or the culture media in water for 20 minutes. And you can also use tyndalization, that is the boiling of water for 20 minutes at atmospheric pressure, and then cool 1 day and repeated 3 times. This is more effective than boiling the media in for 20 minutes. In aseptic techniques, it's very important in microbiology lab, because that is the method to prevent introduction of unwanted microorganism from the environment into the culture media, so that it will not contaminate our culture media. We want to perform the aseptic technique to isolate or to manipulate the specific bacteria or fungi that we want to culture only in the culture media. I think that's it for the brief introduction to the lab 1. You're going to watch the second video for the briefing on the protocol for the preparation of media and also the experiment of the aseptic techniques. Okay, goodbye for now.